Hello and welcome to this webinar. My name is Kristina Lind and I work at the Tata Biocenter in Gothenburg, Sweden. During this webinar, I will talk about different problems that can occur during qPCR, how the problems can be identified and what to do to solve them. I will start talking about problems that can come up and be identified during the qPCR run. I will then talk about problems concerning inhibitors, and in the end about problems with the RNA and the reverse transcription step. So we start with the qPCR assay. The first thing you need to do after having run a qPCR experiment is to do a background subtraction. The curves may have different starting fluorescence, and to be able to compare them, they need to start from the same baseline, like we have here. When the curves are background subtracted, you need to examine the shape of them. Are the shapes of all curves similar, or do any of the curves have a different shape? If such a curve is identified, you need to be careful of its use in your quantification. One reason for strange shape of an amplification curve can be wrong background subtraction. In this case here, in the lower graph, you see a couple of curves from these cyber-green-based reactions that are a little tilted. The fluorescence in the end is decreasing instead of being constant. By looking at the curves without background subtraction, the raw data in the upper graph, you can see that the curves there had a normal shape and that this artifact with the tilted curves has been introduced during the background subtraction. In this case, too many cycles have been used for baseline subtraction. That is, even cycles after the fluorescence has taken off and is not to be considered background anymore. In some real-time qPCR instruments, the number of cycles to use for background subtraction can be changed manually if this kind of problem has occurred. So make sure that the range that is used is only cycles before the inflection point. So if we change the background subtraction, we can get normally shaped curves and accurate quantification. The shape of the amplification curve can also tell you about other events that might have occurred during the PCR. If you see a curve like this, with a sudden jump of the background fluorescence, it can have been a bubble that has burst in the well. This does not have to interfere with your quantification if you are able to make the background subtraction such that it is the cycles after the jump that are subtracted to the zero level. Another very important thing to look for are curves with a different slope in the exponential part. A different slope of the curve implies that you have a different efficiency of the PCR. If the curve is not as steep as the other curves, the efficiency is lower. The DNA molecules are not amplified with the same efficiency, and thus the curves will be delayed and you will get a higher CQ value. This will look like you had less of the DNA in the sample in the beginning than you actually had, and thus make your quantifications wrong. I will talk more about inhibition in a while. This shape of the curves that you see on the right curves here can also occur. Here, the first part of the amplification curve looks normal, but instead of going into the plateau phase, the curves goes into a linearly increasing curve. This behavior with asymmetric amplification may occur if there is excess of one of the primers that after a while, when the other primers has finished, continues the amplification of just one of the strands. Normally, if you have made a common master mix, all your samples will show this behavior. If the amplification curve has normal shape in the beginning, where the CQ value is determined, the quantification will not be disturbed, and the curve can be used. When running qPCR with an assay with dye-based detection, you may get an amplification rising also in your no template control. If this is not from contamination of DNA molecules in your master mix, it probably is from primerdimers. Primerdimers are short, unspecific products that are formed from the primers. If the sample has no target DNA or very low amount of target DNA, it is more likely that those unspecific products are formed than if you have a lot of target DNA. If running a dye-based assay, the primerdimers can be identified by running a melting curve after the PCR run. The melting temperatures of the DNA products in the tubes will then be revealed, and different products can be identified. The primerdimers are often rather short and will thus have a low melting temperature compared to the longer specific product from the positive PCR run. More than one peak in the positive sample tells you that you have more than one product that gives the signal. In this case here, we see that the negative control has a strong peak with a melting temperature of about 74 degrees Celsius, while the positive sample has its major peak at around 80 degrees Celsius and a smaller peak at around 74. So here we actually have some primerdimers in the positive sample that also contributes to the fluorescence signal. This may have given the amplification curve an earlier CQ value than it should have had without the primerdimers. We do not recommend to use a CQ value from such a curve for quantification. The assay is not sensitive enough to manage this low concentration of target DNA, as this obviously is. It is important to remember that primerdimer problem also exists when running probe-based qPCR assays. With probe-based assays, the primerdimers will not be detected by the probe, but they may still interfere with the amplification and affect the results. If you have problems with primerdimers in a dye-based assay, the dyes will also bind to the primerdimers and give fluorescence signal. This may give a higher CQ value of low concentration samples than would be expected. As you see here in the dilution series, the points in the standard curve is not following a linear behavior at the lower concentrations, and thus it will not give correct quantification. If we look at the estimated efficiency from this straight line curve that has tried to fit the standard points, we see that it is above 100%. If you have an efficiency above 100%, check the fitting of the standard curve and see if you have a problem with primerdimers. You should of course not try to quantify anything with the standard curve fit like this. You should not quantify samples that are outside the linear range of the standard curve. So you first need to remove the points with too low concentration so that your straight line fitting can be made correctly. If you run a probe-based assay, you will see a different behavior compared to the dye-based assay. The primerdimers will not be detected by the probe, but if you have too high amount of the primerdimers, they will affect the efficiency of the amplification of the correct target sequence. There will be a competition between making primerdimers and amplifying your target. You will then have amplification curves that comes later than they should have or that do not show up at all, and it will look like you had lower amount of the target DNA in the sample than you actually had, thus wrong quantification. In the standard curve, you may get missing points in the lower range, and also here you should be careful with samples that come within this region, which will be outside the quantification range. When running a probe-based assay, it can be difficult to know if your samples contain primerdimers or not. Normally, you do not have the option to run a melting curve with just the probe. Therefore, we have developed a DNA binding dye at Tata Biocenter called BoxTow that can be used in combination with FAM-labeled probes to measure the fluorescence during a melt curve analysis. BoxTow has other fluorescent properties than FAM and is analyzed in another channel, the Joe or Vic channel, thus not interfering with the quantification that is performed in the FAM channel. The dye is added to the master mix and its fluorescence is used to analyze the melting after the PCR. As you can see here, the probe is not detecting any primerdimers. The red curve, which is the NTC, is not crossing the threshold. However, when we look in the melting curve measured on the Joe or Vic channel, we clearly see a peak also from this reaction. The positive sample, the blue curve, does not contain any primerdimers though, and that is good. If you have several peaks or new peaks in your melting curve analysis, you should also analyze the PCR products on gel. Here you will see if you have a specific amplification or if unspecific products are interfering your analysis. In this case here, we have analyzed several different new primer pairs to find the best couple. In some lanes we can see both several longer bands that are unspecific products and some shorter that may be primer dimers. The primer pairs that give several bands are of course omitted. A way to reduce unspecific products and to get as high efficiency as possible is to optimize the annealing temperature. A few qPCR instruments have the possibility to run a gradient of the annealing temperature, and then different annealing temperatures can be evaluated in the same run. Otherwise, consecutive runs using different annealing temperatures can be compared. Here we have tested annealing temperatures between 65 and 55 degrees Celsius. What we can see here is that if we have too high annealing temperature as 64 or 65, we will not get any amplification at all. With decreasing annealing temperature, the amplification efficiency is decreasing. To reduce the possibility to get unspecific amplification, it is often good to choose as high temperature as possible that gives a high efficiency. In this case, around 59 degrees would be optimal. Now I will talk more about inhibition and how to test for inhibition. Inhibitors are factors that disturb the enzymatic reactions that we use in our experiments. Inhibitors can come from the biological sample itself or be introduced from the extraction procedure. They can affect both the reverse transcription step and the qPCR step. Sometimes the inhibition is so strong that you will not get any reaction at all. But it can also be that it is just partly inhibiting, lowering the efficiency of the reaction. This can be more difficult to detect, but still it can give you erroneous results if you are not careful. Here are some ways to detect inhibition: by doing serial dilution of a biological sample, estimation of the PCR efficiency from the slope of the amplification curve, or with an amplification control or spike. If you do a serial dilution of a biological sample, you will see if the results from the dilution follows a linear behavior. If it is not linear, that is, if the first curve comes closer to the next dilution than expected, it might have been inhibited. This behavior is possible if the inhibition is of a kind that can be diluted away. When you know this, you can dilute your samples to a sufficient level so that the inhibition is not affecting, and then analyze your samples. To check your sample this way is not possible if you have too low concentration and small samples. If the PCR reaction is inhibited, you may also have a different slope of the amplification curve, as I have mentioned before. This can sometimes be detected by visual inspection. You can see here that the aqua-colored curves have a different shape and slope than the other. You should be careful and remove curves with wrong slope, as they will not be accurately quantified because they will have a higher CQ value than they should. Another way to see inhibition, both in reverse transcription reactions and in qPCR, is to include an amplification control, which could also be called a spike. The spike is an RNA or DNA template that you add to your samples. To test for inhibition in the reverse transcription step, RT, you add an RNA spike after your sample extraction but before the RT. If you want to check your qPCR assay for inhibition, you should add a DNA spike before the qPCR step. At the same time, you have the spike sequence at the same concentration also added to a control sample or just in water. After you have run the qPCR, you compare the CQ values from the spike analyzed in your sample matrix and the one that was added to the control sample or water to see if the sample has affected the efficiency of the reactions. If the CQ value of the spike in the biological sample is higher than in the control sample, there is some kind of inhibition. Specially designed spike assays both for RNA and DNA are available from Tata Biocenter to simplify the assay setup for you. To help you evaluating assays and the results, there are also commercially reference RNA, cDNA, and genomic DNA available from different organisms. Let us now continue and talk a little bit more about RNA and the reverse transcription reaction. RNA as such is a much more sensitive molecule than DNA and can easily be degraded or modified by different means. If the RNA is degraded, it can of course affect the quantification of the original number of RNA molecules. RNA can, for example, be degraded by different physical and chemical factors, for example heat, UV light, and different chemical contaminants that are present in the sample or introduced during the processing. Another very efficient way to degrade RNA is by RNases. If these kind of enzymes come in contact with the RNA, it rapidly degrades the RNA in small pieces. When analyzing RNA with qPCR, it is very important to make an effort to avoid steps that may degrade the RNA, and also to check the quality of the RNA prior to analysis, to make sure that you measure on a proper sample. One way to check the integrity of the RNA is to separate the RNA in the sample by size. This can, for example, be made on the Experience system from Bio-Rad or the bioanalyzer or tape station from Agilent Technologies. What you will see is mainly bands from 18S and 28S ribosomal RNA. In a high-quality RNA, you have two sharp bands or two sharp peaks in the electropherogram reflecting intact 18S and 28S ribosomal RNA. Degradation fragmentizes the ribosomal RNAs, giving rise to high mobility fragments that appear as a smearing gel and a broad, fast migrating peak in the electropherogram. From the analysis you can get a value that gives you an estimation of the degradation, for example an RQI value or a RIN value. A high RQI or RIN value indicates good quality RNA and a low value a degraded sample. A drawback with this method, though, is that it mostly describes the degradation of ribosomal RNA that may be a bit different from the degradation of your mRNA. However, as an estimation, it can be a useful method to apply. An alternative approach is to test the RNA integrity on mRNA level. One strategy is the 3'5' assay. To do this, the RNA is reverse transcribed from the poly A tail with oligo-DT primer. The same transcript is then measured with a PCR assay designed to amplify a segment near the 3' end of the transcript, and an assay that amplifies a segment near the 5' end of this transcript. Optionally, a third segment in the middle of the transcript is also amplified. If the mRNA is intact, the three assays should give essentially the same yields. If some mRNA molecules are degraded, only those that are intact will produce full-length transcript and can serve as a template for the 5' assay, which is far away from the poly A tail. The 3'5' expression ratio thus reflects the fraction of intact mRNA. The 3'5' assay is mainly suitable to assess the integrity of chemically degraded RNA. When it comes to the R2 reaction, we have already talked about the problems that can occur with inhibition and how that can be evaluated. Another thing to be careful about is when you want to analyze very small samples and samples with low concentration. You may then get problems with not being able to detect the lowest concentrated samples. To help reduce this problem, Takara has in their RT kit a buffer called Easy Dilution Buffer that can be used for the samples and contains agents that reduce the loss of your precious RNA. In these graphs, we have a comparison of dilution series of RNA from 500 nanogram per reaction down to 0.16 nanogram per reaction. In the left graph, the RNA was diluted in water, and in the right it was diluted in easy dilution buffer. In the reactions diluted in water, the low concentration samples get a much higher spread, comes later, and therefore gets into a region where primer dimers affect the amplification signal. The reactions diluted in easy dilution buffer have a higher linearity and a good replicates even down to very low RNA concentrations. To summarize what has been said in this webinar, be careful and check the shape of your amplification curves and identify outliers. Identify if you have a problem with private dimers or not. This can be important both for probe-based and dye-based assays. Reduce unspecific products and get high efficiency by optimizing your annealing temperature. Be careful identifying if your sample contains inhibitors. Be careful with your RNA and make sure that it is high quality. And in the end, use a carrier or, for example, the dilution buffer from Takara in your R2 reaction if you are working with low concentration samples. Thank you very much for your attention.